Good morning, students. Last time, you remember we were discussing the genetic disorders and we discussed the Mendelian disorders in detail. Now, today, we will take up the chromosomal disorders. The chromosomal disorders are caused either due to absence or excess or abnormal arrangement of one or more chromosomes. Now, these chromosomal disorders can be of different types. It can be aneuploidy. Now, what is aneuploidy? Aneuploidy is the addition or deletion of one or a few chromosomes from the usual diploid set of chromosomes. Sometimes the two homologous chromosomes of a particular pair fail to separate in meiosis 1, or two sister chromatids fail to separate in meiosis 2. Now, this is what is known as non-disjunction of the chromosome, and it results in the gamete receiving either both the chromosomes of the pair or without the chromosome of that particular pair. So, an egg with two homologous chromosomes, let's say that these chromosomes were X and X, that is the sex chromosomes. It fertilizes by a sperm with a homologous chromosome either X or Y. So, what will be the resultant chromosome number in the offspring? In this case, it can be XXX or XX and Y, because there are two XX from one gamete due to non-disjunction of the chromosome. Okay. So, union of an egg without an X chromosome with an X carrying sperm produces an individual having only a single X chromosome. This is a sterile female with many male features. Now, the presence of a single copy or three copies of a particular chromosome has different names. If there is only one copy of the chromosome in the diploid set of chromosomes, then it is known as monosomy, that is 2n minus 1. And if it has an extra chromosome, then it is known as trisomy, that is 2n plus 1. An aneuploid having 4 copies of one chromosome is known as tetrasomic, that is 2n plus 2. Right? Now, an aneuploid lacking a pair of homologous chromosomes is known as nullisomic, that is 2n minus 2. It does not have even a single copy of that homologous pair of chromosome. Nullisomic. Is that clear? Monosomic, if there is only one of the sister chromatids, that is of the homologous chromosome, there is only one. Trisomy, if there is one extra chromosome. Tetrasomy, if there are two extra chromosomes of the same pair. And nullisomic, if none of the chromosome of the same homologous pair is present, that is, it is completely absent. These are the terms which are given to the different deletions or addition of chromosomes. That is a single chromosome. Now, if we come to the set of the chromosomes, that term is known as polyploidy. When we talk about the chromosome number or the sets of chromosome numbers in an individual or in a particular cell, we start from haploid, that is one single set of chromosomes, that is known as haploid. If there are two sets present, then it is known as diploid. If three similar sets are present, that is triploid, and if 4 sets are present, that is tetraploid. If 5 sets of the similar chromosomes are present, pentaploid, then hexaploid, but a general term given to it is a polyploid. So what is polyploid? It is the addition of one or more complete sets of chromosomes or genomes. Right? This means that it can be that sometimes in meiosis 1, all the synapsed homologous chromosomes, they fail to separate and pass on to one pole of the spindle only. This will produce a diploid gamete instead of a haploid gamete. And a diploid gamete, on being fertilized by a haploid gamete, will give the triploid offspring having 3 sets of chromosomes. And if this diploid gamete is again fertilized with another diploid gamete, then it will produce a tetraploid individual which carries 4 sets of chromosomes. Now the polyploids, they can be either from the same species or from the different ones. So broadly speaking, polyploids can be autopolyploid or allopolyploids. Now what are autopolyploids? Autopolyploids, they are due to the increase in number of chromosome sets of the same species. This means that this is an intraspecific polyploidy, right? So autopolyploids are intraspecific. And if we talk about the allopolyploids, it develops in a hybrid between two species by doubling of chromosome sets. For example, capital A and capital B are the homologues or the sets. They will become capital A and capital B. That means doubling of the chromosome sets. So this is an interspecific polyploidy. The allopolyploids, they have multiple sets of 2 different genomes. They behave as a new species. For example, the popular wheat, the hexaploid wheat, tetraploid American cotton. And a classic example of allopolyploidy is the new plant species known as Raphanobrassica. Raphanobrassica was produced by a Russian genetist. You know what he did? He crossed 2 plants of different genera, that is the radish, Raphanus sativa, and cabbage, Brassica oleracea. So this cross was between radish and cabbage. And each of them had 9 pair of chromosomes, that is 18 chromosomes. The hybrid produced were diploid, having 9 radish and 9 cabbage chromosomes. You know, when we produce these allopolyploids, they are usually sterile because the chromosomes are from different plants or different species. But in sterile hybrids, if chromosome doubling is done, either it occurs by naturally by mutation or we double the chromosome in the laboratory using colchicine, because colchicine suspends this spindle formation in mitosis and it results in chromosome doubling. Right? So these plants then bred true. You know, one more interesting part of this experiment is that in Raphanobrassica, although it is a great achievement, but you know what the scientist thought? He thought that by making this hybrid or an allopolyploid, what will be achieved, that the roots will be of radish and the shoot will be of cabbage. So this means in the same area we will have double benefits. We eat the root of the radish and we eat the shoot of cabbage. So we will obtain both these in the same plant. But nature, although made a hybrid, but the result was opposite. That is, it had the roots of cabbage and the shoots of radish. Right? So, although it is a landmark, a great achievement in making a hybrid, but still what was thought was not achieved. So, nature does its own selection, right? I hope this much is clear to you. Now, we'll discuss a few examples which occur due to chromosomal disorders, that is, the ones we have to discuss according to your syllabus, that is the Down syndrome, Turner syndrome, and Klinefelter syndrome. We'll take these up one by one. First of all, we'll take up the Down syndrome. Down syndrome was first reported in 1866 by Langdon Down. And what are the symptoms of this syndrome? It is characterized by rounded face, broad forehead, a Mongolian type of eyelid fold, flattened nasal bridge, permanently open mouth, projecting lower lip, protruding tongue, short neck, flat hands, stubby fingers, creased palm, congenital heart disease. So the victim has little intelligence due to the malformation of the brain. Heart and other organs may also have deformities. Gonads and genitalia are underdeveloped. Very few victims they live to maturity. And those who attain maturity produced Mongolian and normal children in equal proportions. Now, what is the reason for this Down syndrome or the cause? What we discussed a little earlier, that is the aneuploidy. So, Down syndrome is an aneuploid trisomic disease. It is caused by the presence of an extra chromosome number 21. So, it is the trisomy of chromosome 21. What happens is that both the chromosomes of the pair 21 pass into a single egg due to non-disjunction during oogenesis in the mother's ovary. So the egg has 24 chromosomes instead of 23, and the offspring has 47 chromosomes instead of 46. Right? So Down syndrome is also known as the 21 trisomy. Now, this incidence of non-disjunction arises in the ovaries of aged women. Therefore, the women above 40 years of age are more likely to produce children having Down syndrome. So, they should have this fetal testing for trisomy of 21st chromosome. Now, what is familial Down syndrome? This syndrome may run in the family. So, that is why the name given familial. So, it arises by translocation of a large segment or whole of the chromosome number 21 to another chromosome. So, in such cases, the total number of chromosomes remains 46, but there is an extra 21 material because this segment of the 21st chromosome is attached to the another chromosome, usually the 14th chromosome. But this form of Down syndrome is rare. That is, the familial Down syndrome is rare. You can see here the extra chromosomes, that is the trisomy of the 21st chromosome, here displayed on your screens. 21st chromosome 3. Now the features you can see here of the Down syndrome. So this is also known as, Mongolism or the Mongolian idiocy. So you can see the different features. We talked about the eyes, the nose, the mouth here. Now next is the Turner's syndrome. Turner syndrome is an aneuploid condition with a single sex chromosome, that is monosomy. So Turner syndrome is caused by X0 genotype. That means there is one X chromosome from one of the gametes and the other gamete has no sex chromosome. Right? This means that this genotype results from the union of an abnormal egg with a normal X sperm or a normal X egg with an abnormal sperm not having any sex chromosome. So the individual has 45 chromosomes, and this means it will have 2n minus 1 chromosomes. So Turner syndrome is the only viable monosomy in the humans. Now what are the symptoms? How can we identify a Turner syndrome? The individual having Turner syndrome is a sterile female with underdeveloped breasts, reduced ovaries, small uterus, loose skin of the neck, mostly normal intelligence and short stature, and with many male characteristics like heavy neck muscles, narrow hips. And such a female may not menstruate or ovulate. Right? And one thing more, this combination, reverse combination, that is Y0 of the sex chromosomes, that is Y carrying sperm without an X chromosome, is not viable. Sometimes we talk about super females and super males also. So what are these? They have extra genotypes, that is super females may have 3x chromosomes, 4x chromosomes or 5x chromosomes. They show abnormal sexual development and one more feature, that is mental retardation. So the symptoms become more severe with additional X chromosome. Similarly for the super males, they can be XYY, they have unusual height, mental retardation, and they have a criminal bent of mind, maybe, most probably. They are aggressive and they overproduce the male hormones because of the presence of 2Y chromosomes. Now you can see the clinical features of this Turner's syndrome. From here you can analyze all the features. You can see in the picture, the photograph, as well as all the marked features. So these are the symptoms, clinical symptoms from where we can identify a Turner syndrome, besides the, of course, the karyotype. Here you see in this karyotype the sex chromosome here. It is only one, X0. Now the last part is Klinefelter's syndrome. Now this Klinefelter's syndrome is an aneuploid condition with 3 sex chromosomes, that is the trisomy. You can see here on your screens XX and Y chromosomes. This is again due to the non-disjunction. When a female will produce two gametes due to non-disjunction, one gamete will carry both the chromosomes or sex chromosomes, that is the X chromosomes, and the other gamete will not have even a single X chromosome. So when these two X chromosomes, it fertilizes with a normal Y chromosome, or as normal X chromosome, X carrying egg combines with abnormal XY sperm due to non-disjunction, it results in Klinefelter's syndrome. Right? So clear? So here it is trisomy. That means it has 3 sex chromosomes, X, X, Y. Now, what are the symptoms? The persons having Klinefelter syndrome is a sterile male with small testes, usually long legs, obesity, sparse, very less body hair, and many female characters such as development of the breast. The victim usually has normal intelligence. The more the number of X chromosomes, the greater is the mental defect, as I told you earlier also in the super females. So here you see the clinical features of Klinefelter's syndrome. So these are the genetic defects which we studied, which occur due to either absence or presence of a chromosome or part of a chromosome, and accordingly they are given the name of aneuploids, polyploids. I hope everything is clear to you. So children, stay home and stay safe. We'll be meeting in the next class. Till then, good day.